plants in those eukaryotes, but we will take a look at the structure of a eukaryotic cell in reference to how an animal cell differs from a plant cell. And we keep in mind one constant all the time. Structure, it's the same thing with anatomy. Structure defines function. And we always want to remember what is the cellularity of a microbial group. What is the cellularity? Because that's going to tell you how that organism will function. And in our 7 groups, which was the acellular one? Viruses, right. They have no cells. No cells. So, interesting to start looking at the history of microbiology. That's what we did last Thursday. We started to look at the history. And we went all the way back to the late 1600s. So I said that if you look at the actual history, then we go from, let's say, 1700 to 2000. It's really a little more than 300 years, but that's all. And a key item there, in trying to remember that, is there was no origin of life theory at the time when microbes began. No origin of life. So the early experimenters were trying to prove how life could begin. Yes? Could I write over a little bit? Maybe because this thing is in the way. Yeah. Okay. I wonder if I can move it. I wonder if I can put it down. Is that any better? No. No? All right. I can carry it around. Sorry. Didn't mean to upset it. It's pretty much anchored here, isn't it? I don't know where it's supposed to go. All right. So that was where our focus began when we started to say, well, there is no origin of life. So these early guys started to think, well, maybe they could make life from non-living objects. And for a long time, that theory of abiogenesis really prevailed. And then we got to the point where people thought, well, living things seem to come from the air. There was a number of experimenters who did little deals. Did you do any experiments over the weekend? Did you make any life from non-life? No? No, I just said you should have annoyed the family a little more. Right? I rely on a lot of stuff that I want to talk to you about. And I do make packages of handouts, so I did get the machine to work. And I want to give you a package of handouts that will take us through to the first test. So I'll ask you to bring these things with you because I'll be referring to these pages as we go along. Are they wonderful? Not necessarily, but they're important in some way. So let me get these out to you. And I do want you to bring them as you come to class, if you don't mind. Otherwise, I've wasted paper. And I know some of them are very light, and I know some of them are a little bit dark, but I'll walk you through that. To get the machine to work is such a big thing. So here's our outline on some of the historical moments in the history of microbiology. And then I asked you to think over the weekend, if you would, about some folks who made important contributions to science. And that's where we'll continue today. We'll finish off this list and we'll add stuff to it. So I'll walk you through the package and then we'll continue along. You're seeing the interesting temperature shifts that we have in these rooms? Pretty amazing, aren't they? Although I see the chairs are in a better arrangement. You're not sitting on top of each other here with a lot of room over there. It looks a little better today. So I'm happy about that. So here's your study pack. And you are?
Leah. 1 and 3. Thank you.
Wow.
Sorry.
So what do we have on the first page? We've got a bunch of those people I started to talk about, and we'll continue that. The second page is when we start to classify the organisms. they group themselves into kingdoms. The third page is a comparative between prokaryotes and eukaryotes. So it's a little jiffy handout you can run around with. The fourth page is what do you already know about the cell and the structure of the cell and how that will define the function. The next page is a silly diagram of a eukaryotic cell, but it may reinforce some of the things that you already know. And it always looks to me like a hard-boiled egg that we cut the top off. The next page is a list of important microorganisms when we start to group them and talk about their morphology. The ones with circles around them are the ones that you need to know a lot about. The next page is a schematic of what some of our protozoan organisms would look like. We looked at some of them today. The next page is what some of our fungal organisms would look like. And the last two pages that have unreadable pictures there consist of 5 disease-producing fungal organisms, the next 2 pages. And I'll talk about them and incorporate them into our learning. So this is a jiffy reference or a handout for us to be talking about. So where did we end off? Did we end off at Jablot? Am I right about that? No, not.
That is.
Reading. Oh, we ended off reading, so we're ready for Jablot. Okay. Well, Rady, I said, may have been credited with starting the scientific method because he did things that were controlled experiments. And he included a lot of variables. When you don't have all the variables covered, you might not be able to get an accurate result. If somebody came in here and did a psychological survey and only had the women fill it out, that would not be valid because it doesn't include males. So that kind of thing is what plays also into science. So we're with Jablot. So what do you think Jablot did? Well, he did some of the repeat experiments that Rady did. And interesting in his interpretation is that he set up experiments that included those that remained covered and those that he exposed to air. So he reinforced the idea that air produces or brings in living organisms. And he also experimented with the use of heat. Well, that's gonna be bad, because now I don't have one for you. Okay, and who are you, darling?
Gray, Trigon.
Okay, this is your handout for the rest of the term. So what I'm saying is this, he created vessels of organisms. He covered some, so the air couldn't get to them. And he left others uncovered and showed that life forms did come from the air. But then he added one more thing. He boiled some of his samples. And he left some of the boiled ones covered, but then opened up the samples that had been boiled, took the covers off, and exposed them to air. His results showed that the boiled ones that remained covered showed no living organisms. And the ones that he boiled and then uncovered did show living organisms. So his interpretation, accurate, was that yes, air did bring living organisms into the materials that he was working with. He was also the first person to take a bunch of hay from the farm area and throw it into a solution of water. And what he ended up making was a broth media that would support life. Didn't know it really at the time, but he made a hay infusion that was able to support life. So, as this happened in the early part of the 1700s, by the middle of the 1700s, along comes John Needham, who essentially repeated Jablot's experiments. However, his variable was that he did not really boil the covered samples long enough for them to remain lifeless. And even though he did not expose them to air, when he tested them, there were living forms in them. And Needham gets credit for finding spores. He didn't know it. But because he didn't boil them long enough, the vegetative forms were not actually all killed because some of them could go into a spore form. When he broke open the vessels, boiled vessels that remained covered, he had life forms, and that's what would happen with a spore. The spore became non-metabolic, and then when the vessel was cooled and came down to the normal temperature, it was able to restore the living